Hello, everyone. My name is Shang-Chen Hong, the Director of Genomics Research Center, Academia Sinica. It's my great pleasure to introduce our technology entitled Repurposing Clinically Available Medicines as Anti-COVID-19 Agents. And this project was coordinated by Professor Chi-Hui Wong, the former president of Academia Sinica, and including 13 teams and myself. As all of you know, the COVID-19 pandemics caused a lot of trouble around the world, and many countries try to develop the vaccine and medicine for the treatment. Although the vaccine is available now, however, how to develop the oral drugs is still very important. And to shorten the discovery times, and it is possible to use the older drugs and for the new purpose of anti-SARS-CoV-2. And if the older drugs can be found, the side effects are already known. So in our center, we have collected more than 2,855 FDA-approved drugs, and also more than 3,000 herbal medicines. And we try to use the cell-based assay and animal test by hamster, and to screen all of these compounds to find out the potential drugs against the SARS-CoV-2. And after screening, and we found the metafluoroquine, nalopinavir, thioguanine. These compounds are effective against the SARS-CoV-2. And it is possible to go for the clinical trial, phase 1. And also, we found out the Purina and the meat are also very effective. And these two herbs are very common, and people often use as a tea and drink every day. And so these two also show very important activity. And in addition to those new discoveries, the remdesivir was also found. And remdesivir was discovered by Gilead company, and it is the only drugs approved by FDA and used in clinic and in hospital. And for the Purina and the meat, and my colleague, Dr. Jiazhong Zhan, and he tried to study different combinations of these two, and also added other components. And he discovered a tea called Su-He tea. And this is quite useful for prevention when he used the cell-based assay and animal test. And this Su-He tea has already been transferred to 4 companies. And for the remdesivir synthesis, and Gilead has already announced their synthetic procedure. But however, when we found the synthetic scheme, very importantly, there is a 1-to-1 mixture of the phosphoramide intermediate, and this intermediate is a 1-to-1 mixture, and need to use the recrystallization to get the pure S-form compound in only 39% yield, and the other isomer cannot be used, which is a very big waste. And also, when Gilead company, they use this Chiral intermediate and to couple with the ribose derived 5-alcohol to generate a phosphoramide compound, in 70% yield. And this compound can be hydrolyzed in 37% HCl and to get the final remdesivir compound in 69%. And the overall yield is 48.3%. So the yield is not very high, and the price is also very high. So we try to reduce the synthetic pathway and also try to reduce the price. And so our strategy is shown here. So we try to use the 1-to-1 mixture of the chlorophosphate-amide compound and to couple with the ribose-derived alcohol in the presence of the developed organochiral catalyst. And we found out the coupling yield is high, and also we can generate the desired S-form isomer in 96.3%, and the other isomer is 39%. And without purification at this step, we can directly edit the PTSA and the methanol to do the hydrolysis and to generate a remdesivir in a one-part manner. And our advantage is shown here. And we already carry out 10-gram scale. And also, we can obtain 99.4 purity after first recrystallization. And also, the overall yield in these two steps is 70% in a one-part manner. And also, we can recover the chiral catalyst in 80% yield. And this technology will be possible for industrial use. And this is our one-part synthesis of remdesivir. And thank you very much for your kind attention.